The overall goal of this assay is to measure and manipulate the male Drosophila mating drive in order to examine motivation in a reductionist system.
This method can help answer key questions in the study of motivation, like what genes and neurons control mating drive, potentially revealing the logic behind the motivational circuitry.
The main advantage of this method is that it provides a high-throughput assay for measuring mating drive that can be used in concert with the powerful genetic tools available in the fly.
At least 3 days before a 2D satiety assay, prepare female flies from a bottle of white 1118 over heat shock hid fly stock. Once 20 to 50% of the pupae have eclosed and there are at least 5 males to propagate the stock, flip flies into a new bottle. Next, heat shock the original bottle in a 37 degrees Celsius hot water bath for 1 hour to sufficiently activate heat shock hid to kill male larvae and pupae. After the heat shock, only females will eclose from these bottles. To prepare the arena for experiments, partially assemble the 2D satiety arena and tighten it with thumb nuts and hex screws. Next, place fresh fly food into a clean microwave-safe bottle and add just enough water to cover the bottom surface. Microwave the food on high for 30 to 45 seconds. Visually check